Hi, it's Ms. Parrott, and this is video number 5 in the AP Biology Equations Explained series. Today we're going to be learning about the chi-square test. Here's the formula. We're going to break that down in just a second. What the chi-square test tells us is if the differences in our hypotheses and what we find in our experiment significant or not. It's going to compare our observed or measured values to our expected or predicted counts. And then it's going to let us know if the variation we see is due to random chance or if there's something else going on. First, let's break down the formula. Chi-square is a statistic. It is not a function. So this square is not something that needs to be undone. This is the Greek letter chi. It is not x. So we're not solving for x like algebra here. I've just seen students do that a lot in the past. So this just is the number that we're looking for, and we're going to find it by plugging in to this formula. O stands for observed values. E is expected values, or what we predict in our hypothesis. And this big sigma represents the sum of all the calculations for each category. We're going to use it with an example in just a second. So chi-square is most often introduced during genetics labs, but it could be used anytime. So let's imagine we're doing a fruit fly genetics lab where we have two heterozygous flies for long antennae. So if big A is for long antennae and little a is short. We can figure out our expected phenotypes by doing a Punnett square. So if you cross two heterozygotes, you should have a 3-to-1 phenotypic ratio of the dominant to the recessive, or in our case, 3/4 long to 1/4 short. Now, let's imagine in our experiment, our flies produce 200 offspring, and of those 200, 160 had long antenna and 40 had short antenna. Now we're going to walk through the steps. So for the chi-square test, right, because again, we're trying to see, because our data did not match what we expected. So is that difference significant or not? First, what we do is we're going to state the null hypothesis. I did a video on that last week. Check back on the channel for more on the null hypothesis. And an experimental or alternative hypothesis. The null hypothesis, or H sub 0, would state that the differences in the observed and the expected counts are due to chance, and it's not statistically significant. The experimental or alternative hypothesis would state that the differences in observed and expected counts are significant. So maybe there's something going on. So the second step is to determine what our expected counts would be. Because we know what we observed already, right? 160 and 40, but we use our Punnett square and our phenotypic ratios. We multiply those by 200 to get our expected counts. So we would expect 150 to be long and 50 to be short. Now it's time to calculate. Here I just have a little chart where we have our phenotypes, long and short, our observed numbers, and our expected numbers. So I'm going to look at my formula sheet and remember that I'm going to do observed minus expected squared over expected. So for long, my observed number is 160 minus my expected number, 150, squared divided by my expected number, also 150. For short, when I plug those numbers in, that's going to be 50. Wait, no, observed is 40, minus 50 over 50 expected, right? And we don't forget to square the numerator. So 160 minus 50 is 10, squared is 100, and then 100 divided by 150. Again, when you're using your calculator, you want to use the one you plan to use on the AP Bio exam. We're going to get 0.67 plus, this is 40 minus 50 is negative 10. Negative 10 squared is 100. 100 over 50 is 2. So 2. And then, right, we're going to take to find the chi-square value, we're going to add those up. Now, say you were doing something where you had 3 groups, then you would have 3 numbers here. Like if you had a choice chamber with a left side, a middle, and a right side, right? You would have 3. But in this case, we have 2. So we're going to add those up to get our chi-square value of 2.67. So what? What does that mean? Now it's time to interpret our results. So our chi-square value is 2.67. The next step is to find the critical value. So on your AP Biology formula sheet, there's a chi-square table. This is a small version of that. But on it, you'll see degrees of freedom. How do we find the degrees of freedom? For that, we take the number of groups or possible outcomes and subtract 1. So for us, we had 2 groups, right? Long and short. So for us, n minus 1 would be 2 minus 1. Our degrees of freedom is 1. Okay? Now, usually an FRQ would tell you what p-value to use, but the p-value, that's this. They give you 2 rows, one for 0.05 and one for 0.10. This is your significance level. Usually you're going to be asked to use 0.05. Now, I'm going to give you the fancy definition and then I'm going to give you the simple definition. The fancy definition for what the p-value is the probability of rejecting the null hypothesis when it's actually true. Huh? Exactly. So think of it like this 0.05 is a percent, like 5%. So this is the chance that you could be wrong. 5% chance you could be wrong, which means you 95% chance that you could be right. So the lower the p-value, the closer you are to accuracy. All right. Now they put a row for 0.1 there. Why? Just to be sure that you know how to read the chart correctly. So if we have a p-value of 0.5, which the question would tell you, and then we figure out our degrees of freedom, the definition for degrees of freedom is on the formula sheet, but it's a little confusing. So we take our number of groups minus 1 and we get 1. Then our critical value, this is the value we're going to compare our answer to, is 3.84. Again, so what? So what is that if our chi-square value, our chi-square statistic is 2.67, that number is less than the critical value, the one we found on using the chart, the data table, then we can make a conclusion. So if our value is less than the critical value, we will fail to reject the null hypothesis. Again, if this wording is confusing to you, check out my video on the null hypothesis. I'll have the link in the description below. So we don't accept the null hypothesis, that there's no real difference, but we fail to reject it. So we're cool with it. Yes, that hypothesis works for us, the null hypothesis. If you get a 160-40, that's close enough to a 150-50 to say that yes, we have 2 heterozygous parents. This lets us know that the difference in the observed and expected values are just due to chance. The differences in our observed and expected values are not statistically significant. So it's not like we've had some mutation happen or that we didn't know the genotypes of the parents and one was actually homozygous. Our hypothesis, our null hypothesis, is supported by our data here. So in conclusion, the chi-square test is a valuable tool for analyzing categorical data in biology. Just remember the steps to clearly state your null and experimental hypothesis, to calculate your chi-square value accurately using the formula, to determine your degrees of freedom, and then use the chi-square table to interpret your results. Coming up next, our next big topic will be the Hardy-Weinberg equilibrium. But later this week, I plan to come out with another video, a quick one that runs through another example of chi-square analysis. Be sure, if you're a teacher, to check out my TPT store, Know and Grow with Gnomes, for practice on all of these AP Bio statistics. And like, subscribe, and comment below. Have a great day.